Kayla Lee, and I'll be talking about the RNA world hypothesis. So somewhere on the early planet, Earth's chemistry was producing random chains of RNA. Once formed, these chains began replicating, evolving, and competing with each other for survival. The main researchers involved were Carl Woese, Francis Crick, and Leslie Ogle. They believed that the RNA could evolve this way, mainly through base pairing with assistance. They discovered that when a long chain of RNA is suspended in cool water with a high concentration of free nucleotides, the chain can act as a template for its own replication, and the nucleotides automatically base pair with their partners in the existing chain. If their backbone atoms form chemical bonds with each other, this is the part that they need assistance. This was part of the challenge in the research because they're not sure exactly how this happened naturally in the wild. So if their backbone atoms form chemical bonds with each other, then a complementary RNA strand is born with the exact inverse sequence of the one that was copied of the template. And then when water is heated, the chains would break apart and become separate strands of RNA again. However, when the chain of RNA was placed in cool water without that many free nucleotides, the chains would fold up and base pair with themselves, resulting in this complex shape with sticky bases pointing outward. This shape is called a ribozyme, which is basically a folded chain of RNA capable of guiding a specific chemical reaction. They each have different functions. Some of them break certain molecules apart while others join them together, and these functions are determined by the ribozyme shape, and the shape is determined by the sequence of the RNA. After several tests, they discovered that some of these ribozymes could even make nucleotides. So they grabbed the successful chains and they used the PCR technique to quickly replicate them. And with slight random mutations, they found a highly efficient nucleotide building ribozyme. So basically, this hypothesis was like the simple idea that the first things to replicate and evolve on our planet may have been chains of RNA. However, this hypothesis is still being researched and there is still a lot to discover. For example, scientists still don't know how the backbone atoms could bind with each other without the special enzymes and the lab techniques that they use in the lab today. Now Vic will talk a little bit about what happened after this RNA evolved.
Hey, it's me, Vic. According to the hypothesis, life later evolved to use DNA and proteins due to RNA's relative instability and poor catalytic properties. Which is this is the RNA. And gradually ribosomes became increasingly phased out. So the ribosome in this picture became phased out through time. Strongest evidence of the hypothesis is revealed that the mechanism central for translation is catalyzed by the RNA and not protein. To summarize the RNA world hypothesis, it's a simple idea that the first things to replicate and evolve on our planet have chains of RNA, which is the basics of RNA world hypothesis. Thank you.